Every night, as you drift away, a hidden tide begins to flow through your brain, washing it clean in the quiet hours before dawn. While you lie still beneath the covers, channels narrower than a human hair open between your brain cells, and a slow silver river of fluid moves through them, carrying away the residue of the day. This is the glymphatic system, a discovery so recent that most textbooks still do not mention it. And tonight, as you rest, it is already at work inside you, gently sweeping the corridors of your mind. Welcome to Let's Learn While You Sleep. Tonight, we journey inward, past the skull, beneath the folds of the cortex, into the silent architecture of the sleeping brain. For most of medical history, scientists believed the brain had no waste removal system of its own. The body had lymphatic vessels, tiny pipes that clear away cellular debris, but the brain seemed sealed off, mysterious, exempt. And yet researchers always wondered, where does the trash go? Where do the discarded proteins, the spent molecules, the byproducts of a day's thinking, where do they all end up? The answer remained hidden until 2012, when a team at the University of Rochester, led by neuroscientist Maiken Nedergaard, peered into a living mouse brain and saw something extraordinary. They saw a river, a vast, branching network of cleansing currents that switches on almost entirely when we fall asleep. Tonight, as that same system stirs within you, let us follow its course together, slowly, softly, into the chemistry of rest. To understand the glymphatic system, we must first understand the strange position the brain occupies inside your body. It is by weight only about 2% of you, and yet it consumes roughly 20% of your energy. Every thought, every memory, every fleeting sensation produces molecular waste, just as a fire produces ash. In the rest of the body, this kind of cellular debris is gathered up by the lymphatic system, a network of fine vessels that drains through lymph nodes and ultimately empties into the bloodstream. But the brain has long appeared to lack such vessels. It sits, instead, behind the blood-brain barrier, a tightly sealed wall of cells that keeps most substances out, and presumably most waste in. For decades, this was simply accepted. The brain, scientists assumed, must clean itself in some quiet, diffuse manner, perhaps through slow molecular drift, or perhaps not very efficiently at all. And yet the problem nagged at researchers, because if waste accumulated unchecked, the consequences would be severe. Proteins like beta amyloid, the same proteins that form the plaques found in Alzheimer's disease, are produced continuously by healthy brain cells. They must go somewhere. They cannot simply pile up year after year inside the most delicate organ in the body. Somewhere in the architecture of the brain, there had to be a hidden cleaning crew. A system patient enough to wait for stillness and powerful enough to flush the entire neural landscape. The first hint came in the 1980s and 1990s, as imaging technology improved, and a few researchers noticed faint, unexplained patterns of fluid movement around blood vessels deep within the brain. These observations remained curiosities, footnotes in larger studies, but they pointed, dimly, towards something not yet named. The story of the glymphatic system is, in many ways, the story of how science learns to see what has always been there, moving quietly in the background, just beneath our awareness, waiting only for the right instruments and the right questions to be revealed. The breakthrough came in 2012, in a laboratory at the University of Rochester Medical Center in New York. Maiken Nedergaard, a Danish-born neuroscientist with a long fascination for the brain's overlooked support cells, was studying glial cells, the non-neuronal cells that outnumber neurons and have, for most of medical history, been considered mere scaffolding. Nedergaard suspected they did far more. Working with her colleague Jeffrey Elif, she developed a remarkable new technique, injecting tiny fluorescent tracer molecules into the cerebrospinal fluid of living mice, then watching through a special 2-photon microscope where those tracers traveled. What they saw astonished them. The fluorescent fluid did not simply diffuse aimlessly. It moved with purpose, flowing in along specific channels that hugged the outsides of arteries, penetrating deep into the brain tissue, mixing with the fluid between cells, and then draining out again along the veins. It was a circulation system, a true plumbing network, hidden in plain sight. They named it the glymphatic system, a blend of two words, glial, for the supporting cells that make it possible, and lymphatic for its waste-clearing role. And then they discovered something even more remarkable. When the mice were awake, the system worked only weakly, sluggishly, almost imperceptibly. But when the mice fell asleep, the entire network surged into activity. The space between brain cells expanded by roughly 60%. Fluid rushed through with new force. Waste molecules, including beta amyloid, were cleared at more than twice the waking rate. Sleep, it turned out, was not merely a state of rest. It was an active maintenance phase, a nightly rinse, a flood through the corridors of cognition that simply could not happen while the brain was busy with the world. This finding, published in the journal Science in 2013, reshaped how researchers thought about the purpose of sleep itself. For the first time, there was a clear, mechanical answer to a question philosophers and physicians had pondered for thousands of years. Why do we need to sleep at all? Part of the answer, it seems, is that the brain needs the quiet, to clean. Let us slow down now and walk through the actual mechanics, gently, piece by piece, so the image can form in your mind as you rest. Imagine the brain not as a solid mass, but as a vast forest of cells with rivers running through it. The rivers begin at the surface, where a clear, watery liquid called cerebrospinal fluid bathes the entire brain and spinal cord. This fluid is produced deep within the brain, in structures called the choroid plexuses, at a rate of roughly half a litre per day, though only about 150 milliliters are present at any given moment, constantly recycling. From the brain's surface, the cerebrospinal fluid enters the tissue itself through narrow tunnels that wrap around the outsides of arteries. These tunnels are not random gaps. They are precisely shaped channels, called perivascular spaces, lined by the long, slender feet of glial cells, specifically a type called astrocytes. Astrocytes are star-shaped cells, their many arms reaching out to touch blood vessels on one side and neurons on the other. And embedded in those arms are tiny molecular doorways called aquaporin 4 channels. These channels allow water to pass in and out with extraordinary speed. They are, in a sense, the valves of the glymphatic system, the mechanism that lets the brain pump fluid where ordinary diffusion never could. Each time the heart beats, the arteries pulse slightly, and that pulsation gently drives the cerebrospinal fluid forward, deeper into the perivascular channels. The fluid is pushed through the astrocyte doorways into the brain tissue itself, where it mixes with the interstitial fluid, the briny liquid bathing every neuron. Together, this mixed fluid then flows outward, picking up molecular waste as it goes, and drains away along the perivascular spaces surrounding the veins. From there, it eventually leaves the brain, joining the lymphatic vessels of the neck and finally returning to the bloodstream. It is a circulation as elegant as the body's blood circulation itself, only quieter, slower, and far more recently understood. Now, the most beautiful part of the story, the part that makes sleep itself a kind of marvel. When the Rochester team compared the brains of mice that were awake to those that were asleep, they discovered that the very geometry of the brain changes between these two states. During wakefulness, the cells of the brain swell slightly, and the spaces between them, the interstitial spaces, shrink. The fluid pathways narrow, flow becomes restricted. The glymphatic system, while not entirely silent, runs at a reduced pace as though held in standby. But when sleep arrives, something remarkable happens. The cells contract. The interstitial space expands by roughly 60% in those original measurements. Suddenly there is room. Suddenly, the fluid can pour through with vigor. The astrocyte doorways open wider. The pulsations of the arteries drive the cerebrospinal fluid deeper than before. The entire brain, in effect, becomes more porous, more permeable, more open to its own internal weather. Researchers are still working out exactly which signals trigger this shift. Norepinephrine, a neurotransmitter associated with arousal and attention, appears to play a central role. During wakefulness, norepinephrine levels are high, and the brain cells stay swollen and alert. As you fall asleep, norepinephrine drops, and the cells relax, allowing the spaces between them to widen. It is as if the brain, having spent the day in tightly packed concentration, finally exhales, softens, and lets the rivers run. Particularly during deep, slow-wave sleep, the deepest, most restorative stage, the glymphatic flow appears to be at its strongest. Studies using advanced imaging in humans have begun to confirm what the mouse experiments first suggested, that the human brain too undergoes this nocturnal cleansing, and that the depth of our sleep directly influences how thoroughly the cleansing occurs. So as you lie here, breathing slowly, the very fact of your stillness is creating space inside your skull, space for the rivers to flow, space for the day to be gently rinsed away. Consider what is being washed away during these long, quiet hours of slumber. Among the many byproducts of a thinking brain, one molecule has drawn particular attention from scientists, and its name is amyloid-beta. This small protein is produced constantly as neurons fire and communicate, a kind of metabolic exhaust from the ceaseless electrical conversation inside your skull. In a healthy brain, amyloid-beta is cleared away almost as quickly as it is made. But when clearance falters, when the system slows or fails, these proteins can begin to clump together, forming the sticky plaques that researchers have long associated with Alzheimer's disease. In 2013, the same Rochester team, led by Maiken Nedergaard, published findings suggesting that during sleep, the glymphatic system removed amyloid-beta roughly twice as efficiently as during wakefulness. Twice as efficiently. Imagine that, a nightly tide sweeping out the very molecules that, left to accumulate, may contribute to one of the most devastating diseases of the aging brain. And yet we must be careful here, for science is rarely so tidy as a single sentence. The relationship between sleep, glymphatic clearance, and Alzheimer's disease is still being mapped. Researchers have observed that people who sleep poorly across decades tend to show higher levels of amyloid in brain imaging studies, and that even a single night of disrupted sleep can raise these levels measurably. But whether poor sleep causes the disease, or whether the early disease causes poor sleep, or whether they feed each other in a slow, unfortunate spiral, this remains an active area of investigation. What is clear, what the evidence strongly suggests, is that sleep is not optional for the brain's housekeeping. It is the time when the doors are opened, the channels widened, the quiet rivers allowed to flow. Without it, the debris of thought accumulates. With it, the brain has its chance, night after night, to reset. And so each evening, as your eyelids grow heavy and the world dims, an ancient cleansing is preparing to begin. Patient, rhythmic, older than memory itself, waiting only for you to surrender to it. Now let us drift toward another remarkable detail in this story: the role of a single cell type called the astrocyte. For most of the 20th century, astrocytes were considered the supporting cast of the nervous system. Star-shaped, abundant, quiet, they were thought to merely hold neurons in place, to provide structural scaffolding, while the glamorous neurons did the real work of thinking. But across the last few decades, this view has shifted profoundly. Astrocytes, we now understand, are deeply involved in nearly every aspect of brain function, including the cleansing flow we have been exploring. Reach out, in your imagination, toward one of these cells. It looks almost like a microscopic star, with many fine arms extending in every direction. Some of these arms wrap themselves around blood vessels, forming what scientists call end feet, small swollen tips that press against the vessel walls. And it is here, at these end feet, that something extraordinary happens. Embedded in their membranes are tiny water channels called aquaporin 4, named for their job of allowing water to pass through. These channels, current evidence suggests, are essential to the glymphatic flow. They permit cerebrospinal fluid to move from around the blood vessels into the broader tissue of the brain, carrying the cleansing tide deeper and deeper. When researchers studied mice genetically engineered to lack aquaporin 4, the glymphatic system slowed dramatically. The fluid still entered the brain, but it could not move through the tissue with the same efficiency. The clearance of waste was impaired. From this, scientists have come to believe that astrocytes, long dismissed as mere helpers, may in fact orchestrate the very rhythm of the brain's nightly cleansing. And so, somewhere within you right now, billions of star-shaped cells are quietly tending to their work. Channels open, end feet pressed against the rivers of your bloodstream, waiting for the moment when sleep arrives and the great tide begins to rise. They are not idle. They are not passive. They are listening, always, to the body's slow signals, preparing for the work that only the night allows them to fully perform. There is something almost poetic in the timing of all this. The glymphatic system, current research suggests, reaches its peak activity during the deepest stage of non-REM sleep, the phase known as slow-wave sleep, when the brain produces those long, rolling electrical waves that look, on a monitor, like the swell of an ocean at night. During slow-wave sleep, neurons fire in vast, synchronized rhythms, rising and falling together across great regions of the cortex. And it is precisely during these slow oscillations that cerebrospinal fluid appears to pulse most powerfully through the brain. In 2019, researchers at Boston University, led by Laura Lewis, used a special form of brain imaging to watch this happen in real time. What they observed was breathtaking. Every 20 seconds or so, in rhythm with the slow brain waves, a great wave of cerebrospinal fluid surged into the brain. A tide synchronized with the electrical breathing of the cortex itself. The neurons would fall silent, the blood would briefly retreat, and into that empty space, the cleansing fluid would rush. Then the neurons would fire again, the blood would return, and the fluid would recede. This rhythmic dance, repeating throughout the night, suggests that the brain is not merely resting during slow-wave sleep. It is actively orchestrating its own purification. The slow waves themselves may be part of what drives the flow. And here we glimpse why deep sleep, that elusive treasure of the night, matters so much. As we age, slow-wave sleep becomes more difficult to achieve. The deep, restorative phases grow shorter, more fragmented. And with them, scientists believe, may diminish some of the glymphatic clearance that depends upon them. It is one of the reasons researchers are so interested in protecting deep sleep across the lifespan. Not for grogginess alone, not for energy alone, but for the silent, essential cleansing that only those rolling neural waves seem fully able to summon. Consider, for a moment, that as you fall into your own deepest sleep tonight, your brain will be conducting this ancient rhythm, wave after wave, hour after hour. Let us turn now to a more practical question, one that may have been forming quietly at the edges of your thoughts. If sleep is when the brain cleans itself, what can we do? What does the evidence suggest to support this nightly tide? The answers, it turns out, are gentle and familiar, the same advice that wise grandmothers have offered for generations, now slowly being confirmed by the careful work of laboratories. First, the simple matter of duration. Most adults appear to need somewhere between 7 and 9 hours of sleep, and the glymphatic system seems to require sufficient time in deeper stages to do its full work. Cutting sleep short cuts the cleansing short. Second, the position of the body during sleep may matter, though this finding is still being explored. A 2015 study in rodents suggested that sleeping on the side allowed for more efficient glymphatic clearance than sleeping on the back or stomach. Whether this translates fully to humans remains uncertain, but the hint is interesting. Third, alcohol, long known to disrupt sleep architecture, appears to suppress glymphatic activity even at modest doses, according to research in animal models. And exercise, particularly regular aerobic activity, has been associated in several studies with enhanced cerebrospinal fluid dynamics and deeper, more restorative sleep. Even the quiet rituals of evening, dim lighting, cooler temperatures, the gentle release of the day's tensions, these all support the brain's natural descent into the slow waves that drive the cleansing tide. None of this is mysterious. None of it requires technology or supplements or elaborate schedules. It requires only the willingness to honor sleep as the essential biological work that it is, rather than something stolen from the productive hours. For sleep, current science increasingly suggests, is itself a form of productivity. Perhaps the deepest form, performed in silence, beneath awareness, by the trillion small workers of your nervous system. And tonight, as you settle into the long quiet ahead, those workers are already beginning to gather, waiting for the signal to begin. Consider for a moment what happens when sleep is shortened or fragmented over many nights. The glymphatic system, that quiet nocturnal cleansing tide, simply does not complete its work. Waste molecules that should have been carried away in the silent hours instead linger in the spaces between neurons, accumulating like sediment in a slow river. Among these molecules is beta amyloid, a fragment of protein that in healthy brains is produced and cleared in a continuous balance. When the balance tips, when production outpaces clearance, beta amyloid begins to clump together, forming the plaques that researchers have long associated with Alzheimer's disease. Studies from the National Institutes of Health, published around 2018, have shown that even a single night of sleep deprivation can measurably increase beta amyloid levels in the human brain. A single night. The implications are quietly staggering. And yet we should hold this finding gently, because correlation is not causation, and the path from disrupted sleep to neurodegeneration is long, winding, and shaped by many factors: genetics, vascular health, inflammation, lifestyle. What current evidence suggests is that chronic sleep loss removes one of the brain's primary defenses against the gradual buildup of these molecular fragments. It is as if a city's sanitation crews stopped arriving each night, and refuse began to accumulate, slowly at first, then in drifts and piles, changing the very texture of the streets. Researchers are now exploring whether improving sleep quality in midlife might lower the long-term risk of cognitive decline, though firm answers remain years away. What seems increasingly clear is that sleep is not merely restorative in some vague sense. It is mechanically protective, performing a kind of biological maintenance that no waking process can replicate. Somewhere in the soft folds of the brain, in the small hours of the night, the glymphatic system performs a task that may determine, in part, how clearly we will think decades from now. And so each night of deep, uninterrupted sleep becomes a quiet investment, a small offering placed against the slow erosion of time. Let us drift now toward a question that has fascinated researchers since the glymphatic system was first described. Why does it work so much more effectively in some sleep stages than in others? The answer appears to lie in the deepest layers of slumber, the stage known as slow-wave sleep, or non-REM stage 3. During slow-wave sleep, the brain's electrical activity slows into long rolling waves, each one taking roughly a full second to crest and fall. These slow oscillations seem to coincide with rhythmic pulses of cerebrospinal fluid moving through the brain. In 2019, a research team at Boston University, led by Laura Lewis, published a remarkable study in the journal Science. Using simultaneous brain imaging and fluid measurement, they observed that during deep sleep, large waves of cerebrospinal fluid washed into the brain roughly every 20 seconds, perfectly synchronized with the slow electrical rhythms of sleeping neurons. It was, in essence, the first direct visualization of the brain's nightly tide. As neurons quieted, blood volume dropped, and into the vacated space, cerebrospinal fluid surged forward, a slow, deliberate rinse. The researchers described it as a hidden choreography, electricity and fluid moving together in a rhythm older than memory. What this suggests, though the picture is still emerging, is that the deeper the sleep, the more powerful the cleansing tide. Lighter sleep, fragmented sleep, sleep interrupted by alcohol or stress or screens, all may reduce the amplitude of these slow waves, and with them the strength of the glymphatic flush. It is a sobering thought, and yet also a hopeful one, because slow-wave sleep is not entirely beyond our influence. Cool, dark rooms, consistent sleep schedules, reduced evening alcohol, and unhurried, wind-down routines all appear to support its emergence. The brain, given the right conditions, returns naturally to these deep waters. And in those waters, beneath the surface of consciousness, the great tide moves through, washing the inner shoreline, carrying away what cannot remain. There is something else worth considering as we move further into this story: the position of the body itself during sleep. It may surprise you to learn that researchers have begun to explore whether the orientation in which we rest can influence the efficiency of glymphatic clearance. A study published in 2015 by Hee-Dok Lee and colleagues, working with Maiken Nedergaard, used contrast-enhanced imaging in rodents to examine fluid transport in different sleeping postures. The findings, though preliminary and not yet confirmed in humans, suggested that lateral sleeping, resting on one side, may allow the most efficient movement of cerebrospinal fluid through the brain, compared with sleeping on the back or stomach. The researchers proposed that gravity, vascular dynamics, and the geometry of perivascular spaces might all combine to favor this position. It is a curious finding, and one that should be held lightly. Human brains are not rodent brains, and what is observed in a laboratory mouse may not translate cleanly to a sleeping person. Still, the hypothesis is intriguing, and it dovetails with what sleep researchers have long noticed: that side sleeping is the most common posture among adult humans, perhaps reflecting some quiet evolutionary wisdom. There is also a separate line of evidence suggesting that side sleeping reduces episodes of sleep apnea, which fragment slow-wave sleep and likely diminish glymphatic activity through that pathway as well. So even if the direct postural effect on fluid flow turns out to be modest in humans, the indirect benefits of better quality sleep on one's side may still matter. What we can say with reasonable confidence is this: the brain prefers conditions of deep, uninterrupted rest. Whatever supports that condition, whether posture, temperature, darkness, or stillness, becomes a quiet ally to the nightly tide. And whatever disturbs it, noise, alcohol, stress, irregularity, becomes its quiet adversary. The wisdom seems almost ancient in its simplicity. Lie down, be still, let the body settle into its preferred shape, and allow the slow waters within to do their patient, invisible work. Let's turn now to something often overlooked in discussions of brain health: the role of hydration in supporting this nightly cleansing process. Cerebrospinal fluid, the medium through which the glymphatic system performs its work, is itself produced from the water and electrolytes circulating in our bodies. The adult human brain is bathed in roughly 150 milliliters of this fluid at any given moment, with the total volume turning over several times each day as new fluid is produced and old fluid is reabsorbed. When the body is mildly dehydrated, this delicate balance can shift, and although the brain is fiercely protective of its fluid environment, chronic underhydration has been associated in some studies with reduced cognitive performance, headaches, and disrupted sleep architecture. The relationship between hydration and glymphatic function specifically is still being explored, and we should not overstate what is known. But the broader principle holds. The body that sleeps well is generally a body that has been adequately nourished and hydrated through the day. There is also a subtle countercurrent to consider. Drinking too much fluid in the hours before bed can fragment sleep through nocturnal awakenings, reducing the very slow-wave sleep that the glymphatic system depends upon. The wisdom, as so often in physiology, lies in balance. Adequate hydration spread across the day, tapering as evening approaches. Other lifestyle factors appear to matter as well. Regular physical exercise, particularly aerobic activity, has been shown in animal studies to enhance glymphatic flow, possibly by improving vascular health and increasing the pulsatility of arteries within the brain. Chronic stress, by contrast, may impair clearance through several pathways, including elevated inflammation and disrupted sleep continuity. Even modest amounts of alcohol in the evening, while initially sedating, suppress slow-wave sleep and rapid eye movement sleep alike, dampening the brain's nightly tide. And so the picture that emerges is one of quiet daily choices, small habits that, over years and decades, may shape the inner shoreline of the mind itself. Consider for a moment the quiet relationship between exercise and the cleansing tides of the sleeping brain. Researchers have observed that regular physical activity appears to support glymphatic function, though the precise mechanisms remain an area of active investigation. In animal studies, mice given access to running wheels showed improved clearance of waste molecules compared to their sedentary counterparts, and some evidence suggests that aerobic exercise may enhance the pulsatile flow of cerebrospinal fluid along the vessels of the brain. The heart, after all, is not merely a pump for the body. It is also the rhythmic engine that drives fluid through every hidden passage of the skull, each beat sending a gentle wave through the perivascular spaces where waste is gathered and carried away. And so the slow, steady cadence of a well-exercised cardiovascular system may, in ways science is still beginning to map, contribute to the nightly cleaning of the mind. There is something poetic in this. The idea that movement during the day prepares the brain for stillness at night, that the rhythm of walking, swimming, or climbing translates somehow into the rhythm of cleansing while we dream. Beyond the mechanical, exercise also appears to influence the chemistry of sleep itself, deepening the slow wave stages during which glymphatic clearance is most active. People who exercise regularly tend to experience more robust deep sleep, and within that deeper sleep, the brain finds greater opportunity to flush away the residues of thought. And yet researchers are careful here, for the relationship is layered and complex, and there are many other factors that shape both sleep quality and brain clearance. Age, stress, diet, the presence of other health conditions, and countless variables we do not fully understand. Still, the convergence of evidence is gentle but persuasive, suggesting that the body and the brain are not separate kingdoms, but rather two expressions of a single living system, each supporting the other through cycles of effort and rest, motion and stillness, exertion, and the long quiet hours of cleansing sleep. Let us now drift toward another quiet influence on the glymphatic system, the role of hydration, that simplest of substances, water, which makes up roughly 75% of the brain by mass. Cerebrospinal fluid, the very medium through which the glymphatic system performs its nightly work, is itself produced from the water and salts of the body, filtered and refined by specialized tissues deep within the brain's ventricles. The body generates approximately 500 milliliters of this fluid each day, a slow and constant production that sustains the cushioning and cleansing functions of the central nervous system. When the body becomes dehydrated, even mildly so, the production and flow of cerebrospinal fluid may be subtly affected, though scientists are still working to understand precisely how chronic low-level dehydration influences long-term brain clearance. Some studies suggest that even modest dehydration can affect cognitive performance, mood, and the subjective quality of sleep, all of which intersect, directly or indirectly, with the conditions under which the glymphatic system performs best. And so the simple act of drinking water throughout the day, not in great gulps, but in steady, mindful sips, becomes part of a much larger story. A story in which the molecules of every glass eventually find their way into the rivers and tributaries of the brain. There is, however, a balance to be struck. For drinking excessive amounts of water immediately before sleep tends to interrupt the night with awakenings, fragmenting the deep, slow-wave phases during which glymphatic flow is most vigorous. Most sleep researchers suggest tapering fluid intake in the hour or two before bed, allowing the body to enter sleep already well hydrated, but not so full that the bladder calls one back into wakefulness. In this way, the timing of something as humble as a glass of water becomes part of the architecture of restorative rest. Across millions of years, our biology has tuned itself to these slow rhythms: drink, move, rest, cleanse, and the glymphatic system is one of the silent beneficiaries of that ancient choreography. And so, as our journey through the glymphatic system pauses for tonight, let yourself sink a little deeper into the pillow. Your brain is doing its quiet work, opening its tiny doorways, widening its narrow channels, letting the slow silver tide move through the forest of your thoughts. Every neuron you used today, every memory you formed, every word you spoke, and every face you saw, all of it is being attended to by this patient, ancient cleansing system that no one even knew existed just over a decade ago. There is something profoundly comforting in that. The body, for all its complexity, knows how to take care of itself. The brain, for all its restless brilliance, knows how to rest. And rest, it turns out, is not the absence of activity. It is a different kind of activity, gentler, slower, washing away what is no longer needed, so that tomorrow can arrive clean. Breathe slowly. Let the rivers flow. Let the quiet do its work. If tonight's journey brought you closer to rest, we will continue exploring the hidden science of sleep in the nights ahead. The chemistry of dreams, the architecture of memory, the gentle mysteries of the resting mind. Thank you for spending your night with Let's Learn While You Sleep. Subscribe and come back tomorrow night. We'll be here. Sleep well.